Okay, this is the Professor, CBMD PhD, and today's Thursday, May 11th, 2023. It is 7:44 and 41 seconds, p.m. Eastern Daylight Time, and today's research, green biomanufacturing. So let's hop to it here. As tiny microbes can brew, big benefits for green biomanufacturing. Date May 8th, 2023. Source: Lawrence Berkeley National Laboratory. As scientists find new route in bacteria to decarbonize industry, the discovery could reduce greenhouse gas emissions from the manufacturing of fuels, drugs, and chemicals. A research team has engineered bacteria to produce new to nature carbon products that can provide a powerful route to sustainable biochemicals. Researchers use bacteria to combine natural enzymatic reactions with a new nature reaction called the carbon transfer reaction. As this work could also one day help reduce industrial emissions because it offers sustainable alternatives to chemical manufacturing processes that typically rely on fossil fuels. What they showed is that they can synthesize everything in this reaction from natural enzymes to carbons inside the bacterial cell. All you need to add is sugar, and the cells do the rest. As carbines are highly reactive carbon-based chemicals that can be used in many different types of reactions, for decades, scientists have wanted to use carbene reactions in the manufacturing of fuels and chemicals, and in drug discovery and synthesis. As these carbene processes can only be carried out in small batches via test tubes and required expensive chemical substances to drive the reaction. In a new study, the researchers replaced expensive chemical reactants with natural products that can be produced by an engineered strain of bacteria streptomyces. And because bacteria use sugar to produce chemical products through cellular metabolism, this work enables the team to perform the carbene chemistry without toxic solvents or toxic gases typically used in chemical synthesis. And this biological process is much more environmentally friendly than the way chemicals are synthesized today. The researchers observed an engineered bacteria has been metabolized and converted sugars into the carbene precursor and the alkene substrate. The bacterium also expressed the evolved P450 enzyme that used those chemicals to produce cyclopropanes, high-energy molecules that can potentially be used in the sustainable production of bioactive compounds and advanced biofuels. They can now perform these interesting reactions inside the bacterial cell. The cells produce all the reagents and the cofactors, which means that you can scale this reaction to very large scales for mass manufacturing. Recruiting bacteria to synthesize chemicals can also play an integral role in reducing carbon emissions. Close to 50% of greenhouse gas emissions come from the production of chemicals, iron and steel, and cement. Limiting global warming to 1.5 degrees Celsius above pre-industrial levels will require several cutting greenhouse gas emissions in half by 2030. Researchers say that while this fully integrated system can be envisioned for a large number of carbene donor molecules and alkene substrates, it is not yet ready for commercialization. And for every new advance, someone needs to take the first step. And in science, it can take years before you succeed, but you have to keep trying. We can't afford to give up. We hope the work will inspire others to continue searching for greener, sustainable biomanufacturing solutions. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.